This afternoon, we're going to take a look at another aspect of genetic engineering, something that has just hit the forefront less than 10 years ago, and that is the phenomenon of CRISPR, or more properly called CRISPR-Cas9. So let me talk about gene therapy for a moment. Gene therapy is the idea that we could fix somebody's genes because they have some kind of a disease or a disorder. And the idea is not new. Around 1990, the first gene therapy was actually performed, and it was performed on people, young people, who had the disease. It's called SCID, Severe Combined Immune Deficiency Syndrome, or you might kind of know it as the bubble boy syndrome. So here's a young man who has this disorder. You basically have no immune system because you are missing one particular enzyme. So the old way was to take a virus, often called a retrovirus, and insert a good replacement gene into that virus. Then this virus would be used to inject its DNA into, in this case, these would be bone marrow cells that would ultimately make a new immune system. So the virus would inject its DNA that had a new gene incorporated into it, into these cells that came from the patient. These cells would then be grown, they've got this new gene in them, and then they would be injected back into the patient after the patient's own bone marrow had been destroyed. And these stem cells would take over and start to grow, and they would now have the good gene in them. With this, as you can see here in this picture, this is not really a retrovirus, but it's a virus where we would insert this gene. And the thing about retroviruses is that when they inject their DNA into the cell, that DNA actually gets incorporated, as you can see here, into the cell's own chromosome. So everything red there is viral DNA. And if we put the good functioning gene into that viral genome, it's going to now enter into the chromosome of that cell. So back here, these stem cells would now have a chromosome that has a little bit of disabled virus in it, but it carries the good gene. And did it work? Yeah, it did, once in a while. However, the negative aspect was that you didn't always know exactly where that gene was going to be inserted. And so it cured a young female patient. She was like 4 years old, and it worked really well. So in the 1990s, they tried it on other individuals, and this is an X-linked disease, so it actually occurs more commonly in boys. And several, like 3 out of 10, in some of the first trials, actually came down with leukemia. And that's because there's no control about where this virus inserts itself. It might be there, it might be there, it might be there. It could be, well, let's say it is very unpredictable. And so it could disrupt a gene that you need and therefore cause leukemia. But new development doesn't have that problem. So it was announced in 2012. You can see here it made headlines in every kind of magazine. I know you probably don't get too many paper magazines these days, but big headlines. So let's watch this 2-minute clip about this particular phenomenon called CRISPR.
Perhaps one of the most monumental technological events in the history of mankind took place in 2015, but it was largely eclipsed by headlines of Caitlyn Jenner, ISIS, and college kids throwing tantrums. CRISPR-Cas9, a game-changing genetic engineering technique that allows scientists for the first time to make the finest changes possible to chromosomal DNA with relative ease. CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, which is a description of how the method works. Cas9 is the specialized enzyme used for cutting the DNA. Though known as CRISPR-Cas9 among biologists, for the sake of the public, it's usually shortened to simply CRISPR. Technically, CRISPR isn't new. The underlying process was first observed in 1987 by Japanese researchers. It's actually a naturally occurring ancient defense mechanism that was found in strep bacteria. Why is this monumental? Because traditional gene editing techniques using embryonic stem cells are time-intensive and sloppy. It consists of injecting altered DNA sequences into embryos and hoping they are incorporated through a rare process called homologous recombination. What makes CRISPR special is that it doesn't involve any finger crossing. It makes it fast, easy, and cheap to move around any genes in any living thing, from simple bacteria all the way up to people. Technical details aside, with CRISPR's unprecedented ability to edit genetic code, it will make a whole new generation of medical treatments possible. Utilizing this technique, medical researchers have already reversed some mutations that cause blindness, stopped cancer cells from multiplying, and successfully edited human T cells to be invulnerable to the AIDS virus. But the benefits of CRISPR don't stop at medicine. Agronomists have also edited mildew-resistant wheat, indicating a possible future where engineered crops could feed an ever-growing population on a planet increasingly affected by climate change. CRISPR is even shaking up ideas about renewable energy. Bioengineers have used CRISPR to alter yeast DNA so that it excretes ethanol, which can sharply curb society's reliance on petroleum fuels. Not without controversy, Chinese scientists are already raising ethical questions with reports that they've started gene editing human embryos. Without the regulatory constraints found in most Western nations, China is poised to become the leading superpower in this arena, possibly fast-tracking the age of designer babies and post-humanity.
CRISPR, as you are starting to learn from that short video, is actually a gene editing tool, very much like a word processor, where you could find and replace a particular item in your writing. Now you can do the same thing in your DNA. So how does CRISPR work? So a little bit of history here. So the first discoveries were just kind of a recognition that there were these repeating segments here in DNA. And that was 1987. Nobody really knew what they were. They noticed that they would repeat and there was a little spacer, and they'd repeat and there was a little spacer. And that's where the name for CRISPR actually came from. They were clusters of them, and they appeared regularly, and they were periodic, and they were kind of short, and they kind of read the same forwards as backwards, and they were repeating in the bacterial genome. But who knows what they meant, or what they were actually used for. So it wasn't until 2005 where the discovery was made to understand particularly what those spacers are. So it turns out that bacteria have another way of defeating viruses. So we talked yesterday about restriction enzymes. This is another way. When a virus infects a bacterial cell and it ejects its DNA, it turns out that if the bacterial cell survives, it will take the viral DNA and put little pieces of it into its own chromosome. And that's what gets in between these CRISPR elements. So how does that kind of work? Well, it took another researcher, Jennifer Doudna, to kind of figure out how the whole thing worked. She discovered that there are other kinds of proteins associated with CRISPRs. And the big one that we're going to talk about is Cas9. So Cas9 is the name of an enzyme. It's a protein. It's a CRISPR-associated protein, basically. And she actually just like a few weeks ago won the Nobel Prize for chemistry in 2020 with another researcher from France for her work using CRISPR. So it is really cutting edge. And she figured out how this Cas9 protein works with these CRISPR elements. So think of Cas9 as a scissors. It's what's going to do the cutting of the DNA. So this little blob right here is kind of the overall shape of the enzyme, and it's what's going to cut the DNA, but it has to know where to cut. So there's a piece called a tracer RNA. We're not going to talk much about it, but the tracer RNA is going to associate with that CRISPR piece. Now it's not the DNA, it's going to be the RNA that's made from it, and the viral RNA. So the cell, the bacterial cell, will make a copy of the CRISPR as RNA, as well as that viral RNA. And now this complex is going to roam around the bacterial cell looking for matching pieces of DNA, because those matching pieces of DNA would mean that the bacterial cell is being infected by that viral DNA. So let's assume that our bacterial cell now has been infected by the virus, and the pieces of the viral DNA have been copied into this CRISPR RNA. And now it's going to associate with, this is all happening inside of our bacterial cell. So now here we have our copies. Those are going to associate with, there's our RNA, with that enzyme. It's kind of like sending out policemen or the FBI looking for the most wanted. So these little CRISPR elements are looking for these viruses. So here's our enzyme, Cas9. It's got that little piece of tracer RNA. We've got the copy of the CRISPR RNA, and then the viral RNA, which has been copied from the viral DNA. It's looking for anything that matches this sequence. And when it finds it, so here this now is representing our viral RNA. When it finds it, I'm in trouble swiping with my iPad here. There's that viral RNA. The Cas9 and the piece of viral RNA are going to match that viral RNA. So now this enzyme, the Cas9, is going to act as the scissors, and it's going to cut. Now, this is my viral RNA. Those scissors are going to cut that viral RNA. Well, if you cut the viral RNA, the virus isn't going to be able to replicate itself, and the bacteria is going to be protected from the virus. The virus can no longer use the bacterium to replicate itself. So the virus has been inactivated. Voila, the bacterial is protected. But something kind of unusual happens. So the Doudna and her French colleague were kind of wondering, does this system work with any kind of DNA? Can you cut this specific piece that you want with other kinds of genes, not just bacterial and virus genes? And they tried it on eukaryotic cells. They could insert the Cas9 CRISPR complex into eukaryotic cells. They could cut genes there as well. And now some researchers got pretty interested. You know, people are publishing their papers. They were wondering if you can do this with human cells, and could you actually do some repair? So let's take a look at kind of what's going on here. So here we have a piece of DNA that's been cut by a CRISPR Cas9 complex. You can see the cut right there. In a eukaryotic cell, the cell kind of says, oh my gosh, there's an injury here. We've got to fix that. We've got to repair that. And what happens in a eukaryotic cell is that any break like this, the cell is just going to fill in and put junk in here. It's going to put in some bases. Doesn't really matter what bases it puts in, but it's going to fill that in. It's going to repair that break. Well, you've now put a bunch of junk in the middle of a eukaryotic gene. What's that going to do to the gene? That's going to mess up the gene. You have what we call knocked out a gene. You've made it non-functional. Well, sometimes you need to make a gene non-functional because the gene is doing deleterious things to your cell. So it knocks out the gene. The other thing that happens is sometimes instead of filling that space in with junk, if there is other DNA around, it will take that other DNA and put it into that break. So if into your cell you placed your CRISPR and you placed some DNA that you want to put in there, then you're going to be able to insert that DNA into that blank space. So let's use an example to see how that would work. Let's look at cystic fibrosis. So cystic fibrosis is a disorder of a membrane protein that is responsible for moving ions across membranes. And it particularly is important in your lungs and in your pancreas. And if you don't have that protein, this transmembrane protein that moves ions correctly across the cells, what happens is you build up mucus, and mucus clogs your airways, and then your little alveoli start to die. So here you see a patient who's got pretty clogged lungs as opposed to a very clear set of lungs over there. So it's just one gene, it's a single gene. We'll call it the CTCR, the cystic fibrosis transmembrane conductance regulator gene. It's got a fancy name. I like to just call it the CF gene. And what we want to do is we want to try to put into that gene a good functioning normal gene. Can we do it? And the answer is yes. We can create well, sometimes a virus, but you can take cells in a petri dish, and into that cell, you can inject the CRISPR-Cas9 complex and the normal CFTR gene, the good cystic fibrosis gene, and CRISPR will find that particular spot. That's the key. You engineer the CRISPR so that it's got the exact piece that matches where you want to put this gene. You're not going to put it randomly. You're going to put it at a specific location. And then the Cas9, the enzyme, is going to cut that particular gene. And since you've also put in the donor DNA, since you put in the correct cystic fibrosis gene, it will now fill in and replace or put into place that good gene. And that has happened certainly in the culture dish. And now people are starting to use it as experimental treatments for certain kinds of disorders. Cystic fibrosis would be one of those disorders. So what have we done with this? Well, actually, we've done a lot. So we're not going to have time to talk about this and this and this and this and this. So here's just one example. Wouldn't it be nice if you bought your mushrooms in the grocery store and they stayed fresh in your refrigerator for much longer than they typically do? Well, one of the things that CRISPR has been used for is to knock out the gene. So that would mean you cut it and then it just kind of replaces it with junk. And that by knocking out that gene, it slows down the browning process. More importantly, the FDA has decided that doing that is not actually considered genetically, won't consider those mushrooms a genetically modified organism because you haven't introduced any new gene into those organisms. All you've done is changed, genes that already exist in there. So it's not regulated like other kinds of genetically modified organisms are that are receiving genes from viruses or bacteria or from a totally different organism. So what about people? So in 2015, the first CRISPR editing human embryos occurred. You might imagine that caused some excitement, and it did. This researcher from China was simply taking embryos that were abnormal from IVF. He was trying to see if he could modify and fix a gene in those embryos that carried a blood disease. He had never intended to put those embryos back into a person. There was no attempt to create a child. But he did finally find some journal that would publish his paper. And the reaction to that was pretty visceral throughout. So it took a while to do that. And then in 2016, this researcher used human adult cells, so these were not embryo cells, to see if he could fix a problem that was causing or preventing the person from eliminating cancer cells? And the answer is yes, he was able to do that. So we've got the first example of human edited adult cells. There we go. But what about embryos that would be implanted into a woman and then raised to adulthood? Has that occurred? Well, the answer actually is yes. And there's some controversy, obviously, around this. So, this gentleman, also from China, was the first individual to gene edit embryos on purpose that would be implanted into a woman. So, and the embryos that he used all came from families where the husband was infected with the HIV virus. And they were going to do IVF so that the sperm could be washed and there would be no risk of infection for those embryos. But the families wanted to kind of fix those embryos and change those embryos so that they would be immune to HIV. And so what this individual did was he inactivated, using CRISPR and Cas9, he inactivated the gene that makes a protein called CCR5 that the HIV virus needs to enter into a cell. So these embryos, who were female, had their gene knocked out, and then these embryos were implanted into their mom. And 9 months later, uh, they were born. Well, he announced his work, and immediate uproar in the scientific community. He was fired. He shared his findings, however, and so now scientists are really thinking about, do we need to totally stop this kind of research? So in 2015, even Jennifer Doudna kind of said, I think we ought to have a moratorium on using this CRISPR with human embryos. In 2019, 2020, she's pulled back on that a little bit. She worries about the valuable research that could be done, but that we should regulate this rather than have a complete moratorium.